Welcome, everyone. I'm Shauna Wolverton. I'm the Chief Product Officer here at Benchling, and we're going to try something a little different this quarter. We thought it would be great to do a year in review, the Benchling and Biotech year in review. Talk about some of the great moments over 2022 with our customers and at Benchling. Super excited to bring in some of my colleagues to help us out. But before that, let's do a little housekeeping. So you'll see in the webinar, we've got some great ways to have you participate today. We have our Q&A tab, so make sure if you have any questions throughout the webinar that you can go ahead and post them there. We'll make sure we get those answered. And this time we've got this place for you to chat, so all of you can talk to each other right here in the webinar. And then we're going to put together a docs tab because we've heard from you that after these webinars, you want more. So we've made a place for you to go so you can get more information about the things we've talked about today. Every day, you are fortunate enough to wake up, maybe enjoy a coffee and a moment of calm before heading out to do your work. And for many of you, that's into a lab or an office space or an office near a lab. And you go about your scientific work day in and day out. And you do that work when the macroeconomic environment maybe is a little turbulent, like it's been recently, in an industry that is inherently uncertain. And in this way, it might not be easy to feel the sort of joy and awe of what we do each day. It's easy to lose appreciation of the enormity of what we're all pushing forward. Yet those feelings are there, right? They're in us because we know that we are working on incredible, life-changing things. We are profoundly lucky, the ones who get to help really fundamentally change the world. You are creating products that improve the human experience, from fighting global warming to saving lives with unbelievably advanced new treatments. There truly is magic in your science. So as we look back on 2022, we want to bring that joy and awe to the forefront. In Benchling's Year in Review, we'll share scientific accomplishments and advances in technology, stories of breakthroughs, big and small, and updates on new advanced tools that are available to make the work easier and faster, because we know that every day matters. Because even in the face of increased turbulence, there has also been tremendous progress, and that is something to celebrate. To put these feats into context, let's take a look at some numbers. Even though the market had some big and unanticipated shifts, biotech is still moving ahead with a strong and growing position. This is driven largely by consumer and patient demand for biotech products in the long term. For example, 65% of pharma assets currently in clinical trials are now driven by biomolecules. This includes the over 2,000 personalized medicines currently in development. It's amazing. The FDA approved 3 gene therapies this year. This includes the very recent announcement for Benchling customer Unicure to treat hemophilia. In addition to changing these patients' lives, this also represents the year that gene therapies matured out of clinical trials and into commercial applications. This all provides wind at the industry's back going into 2023, where upwards of 15 different cell and gene therapies are expected to undergo review and approval in the US alone. Although biotech IPOs slowed dramatically this year, it's worth noting that the biotech industry still raised more funds than any other sector. With 8 public offerings in 2022 and 68 VC rounds raised in just the last 5 months, this continued funding increased the likelihood that treatments for neurodegenerative conditions, cancer, and serious infectious diseases could be addressed in novel ways in the very foreseeable future. Biopharma M&A deals picked up, and Benchling was excited to see so many of our largest enterprise customers partnering or investing in some of our most innovative high-growth customers. All this means that the biopharma industry is still anticipating a 7.4% compound annual growth rate for 2022. We also saw the consumer hunger for healthier and more environmentally friendly alternatives to animal and chemical-based food continue to push the growth rate of the plant-based market to 12% this year. We're honored that scientists and companies, including Joywell and Turtle Tree, are bringing these healthy products to consumers using Benchling. Other consumer products like the clothing that Bolt Threads creates from mushrooms continue to lead the way too. And as agritech customers like Syngenta will attest, the demand for high quality, higher yield per hectare and disease and drought resistant plants continues to drive growth for the global engineered seed market. Over at Benchling, we were busy growing through change too, delivering new software and innovation and supporting our customers' success. After 10 years in market, Benchling passed the 1,000 customer milestone this summer and is supporting over 500,000 users, 350,000 in academic and research institutions, and 165,000 in enterprises. A tremendous number of biotech startups also became Benchling users this past year. Companies like RNA Therapeutics, Pairwise, and Anagenics represent the most ambitious and fast-growing biotechs in the industry. As a result, we've seen that 25% of biotechs that have IPO'd in the last few years have been Benchling customers. Benchling's users can now be found doing incredible work in 16 countries around the world. As a leader in the massive drive toward cloud computing for biotech R&D, Benchling continues to support existing use cases while also broadening the type of work that can be done on the platform. We launched 2 new applications this year, studies and workflows. In addition, we released over 40 significant new features. To keep up with the market, Benchling itself has grown 25% in 2022, welcoming 225 new Benchlings, joining our mission to unlock the power of biotechnology. Let's take a look at what products this team has delivered this year. From tools to design complex and conjugated biomolecules to functionality that expedites early development and integrating in vivo studies, Benchling continued to innovate with biology's most advanced software. And it was an absolute thrill to partner with all of you, our customers, as we built these offerings. Our team is always pushing ahead of what the industry needs to help pave the way for next-generation scientists to work even more effectively, all while responding to the current needs, delivering functionality to improve your everyday tasks. Let's spend some time on what these big driving trends are, what we hear in terms of industry needs, and how Benchling has responded with new capabilities. Though there were many forces at work in 2022, we wanted to reflect on 3 specific shifts we saw this year. Taking advantage of advances in targeting with RNA, connecting research, development, and manufacturing data and workflows to accelerate timelines, and working across sites, teams, and partners with new permanent approaches to collaboration. To jump into the breakout role of RNA, let's check in with Gaurav. Gaurav?
Thanks, Shauna. So I'm glad we get a chance to talk about RNA therapeutics, including RNAi, antisense RNA, and aptamers. Because of the recent hyperspeed of research and development, manufacturing at global scale, and worldwide rollout of the mRNA-based COVID vaccines over the last 2 years, it seemed as though RNA therapeutics were already a mature, almost common drug modality. It seemed that the biopharma industry simply needed to do the same thing that they have been doing before this new SARS-2 virus. In reality, the evolution of RNA therapeutics required a deep understanding of the underlying science first. 8 Nobel Prizes have been awarded to scientists working in this field, contributing breakthroughs in science that have expanded our understanding of RNA and its role as a therapeutic. This research has spanned decades, where scientists have worked on increasing the potency, stability, durability, and precision of these biomolecules. This long, productive history, combined with the breakout success of the COVID vaccines, provides the perfect launchpad for RNA therapeutics to take off. And hundreds of biotech companies now have begun to focus on RNA therapeutics, with over 1,300 RNA drug candidates in the industry's pipeline today. However, the industry is still saddled with challenges, in part related to the complexity of these biomolecules, and partly because the science has outpaced the tools to support it. Over the last couple of years, we've heard from scientists working on RNA that iterative oligo and sequence design is slow and inefficient, as these complex biomolecules often require edits to the sequence and structure. Secondly, the lack of clear and standardized syntax makes it difficult to work with vendors and external partners. Also, collaboration across internal team members can be cumbersome, even if trying to work with their own syntax. And finally, connecting oligo and sequence design with actual characterization data is time-consuming. Researchers need to continually understand the modification implications by connecting molecule design to actual assay data. Because Benchling is a nimble platform, we knew we could create tools specifically to address these challenges, and quickly. So this year, we went ahead and released mRNA design, visualization, and analytics tools. In case folks haven't seen what this looks like in action, let's go over to Wendy for a demo of the RNA capabilities. Wendy, while you're sharing the new RNA capabilities, could you also show us what sequence design and molecule entities look like in Benchling?
Thank you, Gaurav. Let me give you a quick peek into the new tools we added this year to support RNA research. This view might look familiar to those of you using the Benchling molecular biology tools, but as you see, this is not representing a DNA, but a molecule of modified RNA. Benchling added new and specific tools to support RNA research, so scientists have an intuitive and easy to use design tool to visualize RNA, design new molecules, which might or might not include chemical modifications. Here on my left, I can see the main sequence. I can see also translation information. I can see information about the annotations, and I can see the details for each of the monomers, all the way to the structural, the chemical structure information of each of the components of the nucleotide. These tools gives teams a common visual and descriptive way to represent and communicate around these molecules. On my right, I can see each of the nucleotides and modifications. In the metadata tab, I have attributes linking this molecule with other entities, such as the DNA, the translated protein, the plasmid, maybe the conjugation on this molecule. This system also can automatically compute this standardized nomenclature to other commercial and commonly used syntaxes, or even a custom defined one. Information that updates live and can be exported and shared with collaborators, CROs, etc. This computation is bidirectional, so you can add new sequences to the system by bringing information in different syntaxes as well. To support the design team, the team that is inside the design process, Benchling offers several ways to search and modify these sequences. For example, I can select some of the nucleotides, and I can change any of the monomers for those nucleotides and substitute maybe the sugar for a different sugar, a monomer of the base of the phosphate. And those changes will apply to my sequence. Or I can select the entire sequence and search for a specific monomer, and substitute that monomer for, again, any other monomer that might be in my library. We have significantly improved our searching capabilities. But since we are talking about RNA, I want to show you a specific case for the search that is related to these molecules. Of course, I can search by name, sequence, annotations, but more specifically, I can use my filters to find molecules, maybe with a particular monomer in a particular position, or a minimum count, a maximum count, etc. This year, we added as well more new tools to support the chemistry teams. We have added a new type of entity to our registration system, a new entity that supports chemical structure information, search, and design. These tools allow the chemistry team to centralize all the relevant information about their molecules and their processes. So here an example, I'm going to give you a quick example of this functionality. So in this case, our modified RNA molecule has a chemical modification. The chemistry team that carries this conjugation process has captured the process in the Benchling notebook. And it's using the Benchling registry now to capture the molecules and to add the visualization to the structure and automate the calculation of biophysical properties. And it's using also the design tool to actually modify, to change the structure and explore the structure of these molecules as well. This year, we added as well more and new tools to support the chemistry teams. We have added a new type of entity to our registration team system, a new entity that supports chemical structure information search and design. I'm going to show you an example. I'm going to show you an example so we can kind of showcase this functionality. This molecule of modified RNA was actually conjugated to a chemical compound with a chemical modification. We added a new type of entity to our registration system, a new entity that supports chemical structure information, search, and design. These tools allow the chemistry team to centralize all the relevant information about their molecules and their processes, and also empowers collaboration between the chemistry, the biology teams, analytical teams, QC teams, etc. The chemistry that carries on the conjugation process has captured the process in the Benchling notebook and is using the registry that now has added chemistry functionality, such as visualization of the structure, as you can see here, and also automated calculation of the biophysical properties and the design tool. To support the molecular design, we have added sketching functionality. The design can be done right here in Benchling. As this new level of information is available now in Benchling, so we can also perform structural search through our database. We can, let me show you here an example. I have an example where I have used the filters, and what I have done is a search, a structural search by sketching this element and letting the system find this element throughout my database, through all the compounds in my database. So we have seen some examples of how this year Benchling has worked really hard on tools to support RNA and chemistry teams. And I hope you had a chance to I know it was quick, but at least have a little bit of information about how this new functionality works. I'm going to pass it back to you, Shauna.
Thanks, Gaurav and Wendy. That was amazing. Before we jump to our next topic, I want to highlight that there are some really easy next steps you can take if you want to take advantage of the capabilities that you've just seen. For RNA design and visualization tools or sequence design capabilities, reach out to your customer success manager. You can also check out the resources tab in the webinar platform for the molecular biology product sheet to share with colleagues who maybe couldn't join us today. Since the small molecule entity registration requires an integration with Marvin.js, the best person to reach out to would be your sales executive. Now, let's check in with Kate, who will talk about the very exciting industry trend of connecting data and workflows across research, development, and manufacturing to accelerate those timelines. Kate?
Thanks, Shauna. The overall pipeline advancement for biologics, including cell and gene therapies, monoclonal antibodies, ADCs, vaccines, recombinant proteins, and many more modalities continued full speed ahead this year. According to a recent global data report, nearly all types of biologic modalities are expected to grow in sales revenue and will account for 55% of all innovative drug sales by 2027. Gene therapies and gene-modified cell therapies are expected to display the fastest growth, with a forecast increase of more than 1,000% from both molecule types in the next 4 years. These projections are based on the overall pipeline view of IP that is starting to move out of the lab and into production. Paired with the FDA's revamped programs for faster review and approvals, these assets are moving more quickly into clinical trials. At Benchling, we've seen a tremendous number of our younger customers going from discovery insight into phase 1 and phase 2 trials, while scaling up and out their operations rapidly. And our large global leader customers are adjusting by partnering or acquiring at a fast pace too, as they focus on new ways of developing and manufacturing these large molecules at scale. Meanwhile, the continually ballooning CDMO market indicates the ongoing demand growth for producing these products is apace. Many companies this year announced their own manufacturing facility plans and groundbreaking. All that is to confirm that the transition from discovery to development is fast and multifaceted. Many bioprocess scientists who are moving these molecules into and through early development are still typically working on a fragmented ecosystem of point solutions like email, spreadsheets, antiquated or homegrown software, on-prem systems, and scientifically oblivious tools like Asana, Jira, or Google Docs. In particular, we hear about the pains associated with underperforming traditional ELN and LIMS solutions. Where traditionally ELNs have been built to support exploratory, hypothesis-driven experimentation, they usually do not have process management functionality, aren't sample-centric, and are too flexible to capture the structured data required in development. And on the other side, LIMS that are created to drive protocol-driven processes in development are typically too rigid to capture and manage the unstructured data found upstream. And that historically ELN and LIMS have been disparate solutions disconnected from one another causes additional challenges. Common issues include knowledge transfer among R&D teams that is overly time-consuming, which strains momentum. Downstream teams lack context for prior work done upstream, which often leads to wasted time and effort rerunning experiments. The need to recreate sample lineage from multiple systems when it's time to file an IND, for example, can be tedious and time-consuming. There's missed opportunity to streamline processes within and across teams. And lastly, we hear from IT leaders that they spend an inordinate amount of time maintaining these legacy systems. That's why innovative R&D teams tell us that they not only want to work together to prevent or reduce these difficulties, increasingly, their success depends on sharing data effectively throughout the R&D lifecycle, which is why Benchling unveiled biology's modern LIMS this year. By taking advantage of Benchling's shared cloud-native platform, both research and development teams can collaborate within the same powerful and easy-to-use digital interface and data model. This promotes working together seamlessly across highly specialized teams, including cell line development, upstream and downstream development, fermentation, and bioanalytical teams, for example. Importantly, it adapts to the needs of today's biotech innovation with regular releases of new process innovation capabilities to take advantage of as your organization and the industry's needs evolve over time. For those who haven't seen Benchling's LIMS in the most recent launches, let's tour some of the key features. Wendy?
Thank you, Kate. We are back in Benchling. I'm going to give you a quick tour through the new application workflows. We'll use an example I believe is relevant to many of our customers. So let's imagine I belong to the production team. As part of the process, we have prepared a new lot of the new drug, and we have captured everything in our notebook, all the details of that new lot, including inventory. So right from here, so the next step would be for us to send a request to the QC team to run some quality control. And we can do it right from here, right from this document, without the need to send emails, to write information on a piece of paper, to go to somebody's desk. So right here, I can initiate that request. And this information, this new application with the information that is transferred from one place to another will allow the teams to stay connected and receive the information and pass the information one to another. So here you can see that this form is capturing the vial, the ID for the lot. So no worry about sending the wrong information to the QC team. And so in many cases, especially for QC teams, so these processes, these steps, the tasks from these requests will be logged and reviewed and accepted. But it will be other cases in which maybe we need to change some parts of that workflow, that structure. And the new flowchart builder in Benchling will allow us to for those use cases. So let's imagine in this case that maybe because of the type of sample, or maybe because I want to try a new process, I don't need a potency test in this case, but what I need is actually an HPLC, or I want the QC team to run an HPLC on my sample. All I have to do is substitute that test that was there for one of the ones that are in my library. And here I have a new configuration of the QC team workflow that will be submitted to that team. So you can see how these workflows can support the research teams and the flexibility they need, but also those teams that need those processes locked down. So as I create this request, so what I can see here is that, yeah, this panel is bringing all the information from the production team. The QC team is going to receive a notification, and the QC team will be able to access, again, all this information right in the platform. This workflow actually receives or is assigned to an ID, and everything belonging all part of this workflow will be tracked under that ID. You can see here too that the sample has already moved to the first of the steps, the first of the first task, in this case, aliquoting. Imagine now that I'm part of the QC team, so I have received a notification in which is I'm alerted that I need to do some testing, and the first step of the testing is aliquoting that lot that was sent by the production team. So I can come here and in this panel, so I can do things like, for example, assign this task to a particular person in my team. I can schedule for a particular day. And right from here, I can execute and initiate this task. Each of these tasks is going to be associated with a templatized document that is going to guide the scientists through the steps of each of the tasks. This opens this templatized document, and I can see here that the information is transferred from the workflow into this document, that I have ways to retrieve more information from the system, and then we are going to process the first part of the action on these tags right here in this document. So something that I'm going to do is actually assign these tubes, assign inventory allocation for these tubes. And something that I have done as well is actually assign sample owners and authorized users to each of the aliquots. I have marked which test will be done for each of the aliquots, and in this information will be transferred throughout the workflow. I can confirm the outputs for this workflow of this task, and I can even set maybe, if I need more information, maybe like urgency of the test. As I complete the outputs, and I submit the outputs, this task will be marked as done, and the process will move through the testing and through the workflow. So again, so now the next teams that are going to run the test will be notified. You can see here how the task, the aliquoting task that was just completed, is marked automatically, and the aliquots are moving automatically as well through the steps of the workflow. Let me show you a complete workflow. Similar workflow, in this case, all the tasks have been completed. Again, both the production team and the QC team have access to all the information, all the actions that happen through the workflow, including the details of each of these actions. So I can easily go into that document in which the HPLC test was done and find all the details of what happened, of the results, all the outputs, etc. As we complete this information, all the information, as I mentioned before, is captured in a structured way in the system. And all the workflows can be explored, and the status of the task can be seen in this easy panel. And so I can easily understand what's going on with all the workflows, or maybe some specific workflows. And if I filter the information, I can even see maybe I want to see all the tasks that are assigned to me. And I can, make decisions of reassigning tasks, or maybe scheduling my own time or my team's time. I hope I give you a good overview of this new application. And I hope I was able to show you how the teams can connect right in the platform with transferring all the information about the tasks, the samples, the results, etc. Back to you, Shauna.
If you're interested in taking advantage of these new capabilities, you'll want to reach out to your sales account executive to learn more about workflows and a customer success manager to begin using the sample control functionality. The Resources tab also contains the Workflows product sheet as well as the white paper on Benchling's modern limbs for biology that you can download. For our last theme, we want to check in with Sean on how the world of R&D collaboration has settled into new approaches to how we work now that the dust has settled on the pandemic upheaval. Sean?
Thanks, Shauna. Although biotech has been working to digitize processes for the last 5 to 10 years, the pandemic certainly did accelerate how organizations collaborate in a more dynamic and connected way. This past year, teams settled into their new organization and data sharing arrangements across teams and sites, finding that they gained significant efficiencies. Data scientists are some of the most in-demand roles in many organizations, where they structure, harmonize, and identify value out of the vast collection of information. There is a more fluid exchange of what's working and what's not. This means sharing within a particular program across research and development, and also across the entire R&D portfolio. We have heard from R&D leadership that they now focus more on utilizing intelligence from academic and industry partnerships. There is a push to use more open source and shared innovation centers to reduce the massive cost of R&D. An advanced example of this has been the collaboration among Arzeta, Twist Biosciences, LabSite, and Tesselligen to share an assembly platform for DNA in order to produce food products and other industry essentials more efficiently. As we know, this frictionless exchange of highly valuable information is not yet commonplace. R&D leaders and tech leaders are still working to remove data from silos and integrate disconnected systems, to increase the ease of collaborating, and to pull the right data at the right time in order to generate truly helpful insights. This work continues to increase how reliably, efficiently, and nimbly organizations can run. At Benchling, we also focused on connecting more data and scientists into a shared digital ecosystem. This year, we delivered integrating in vivo studies management into Benchling with a new studies application, integrating with more key partner systems like Quartzy and Cytara, setting up processes for reviews and approval of entries, and making all of this data even more searchable faster. Let's take a look at what these new capabilities look like.
Thank you, Sean. Let me show you what studies looks like and how scientists use it to streamline, centralize, and standardize in vivo studies. I'm going to give you a quick tour, but please don't hesitate to ask for a full demo and if you want to know more about this part of Benchling. So here's my main dashboard with all the studies that I actually belong, I have access to. I'm centralizing all the information in one place and all information about all the studies and each of the studies in particular. So you can see I'm inside in one of the studies and I can see all the information about the animals in that study, the cages, the attachments, the samples collected on those animals, observations, the treatment groups, everything is here. For each of the animals, I have not only where they are housed, but also metadata that all types of metadata. And animals can be added to the study and specifically to different groups using the in-platform randomization application. So we have done it already here. This would be a process that you can do right in the platform for each of your studies. Capturing information and capturing data for your studies is pretty easy and streamlined, and you can do it in different ways. You can maybe go to a particular animal, and of course you can add new measurements for a particular day as you go along the study. There's also ways for you to import the data from some tabulated document that might come from a CRO, might come from an instrument. But also, studies has designed a way for you to streamline the data collection. The data collection, this part of the platform is going to allow you to go animal by animal and identify those animals in different ways and actually create this workflow in a way that all the data is collected in a stream way. So I think the best way for me to show you how this works is with a short video that I have here. So you can see scientists is scanning the tag in each of the mouse. The system recognizes that tag, and the scientist is collecting information with that Bluetooth caliper. The information is entered automatically and is collecting the measurement, the weight, the body weight of the animal on that scale that is connected to the system through USB port. So this is obviously streamlining the data collection and giving the scientists tools to avoid errors to make this process much easier. So studies is now part of Benchling, and we understand that important part of the process is connecting those teams, connecting the team, the in vivo team, with other teams that actually perform assays and collect data, maybe on the samples that are captured of those animals. So I'm going to show you an example of that integration, that connectivity between the teams. So I'm going to go here to my sample and my samples. I'm going to go to this particular sample that comes from this particular animal. As soon as the in vivo team captures the information of this sample in studies, that information is also automatically captured and in the Benchling registry. So I'm here now. This is a view of the registry in Benchling, and you can see here the information for the sample has been automatically be added to the registration system under like animal samples. This information includes the animal ID, includes the group, the study, all the information that was collected by the in vivo team. And connected to other teams, like let's see now that other teams have done some tests, some research on that particular sample. Here I have two examples of those actions, like the histology team and maybe the team that run the qPCR, and all the information again centralized in one system. And no matter if I'm a part of the analytical team or the in vivo team, I can always access this information and all the details of this particular experiment. And not only that, actually new to Benchling this year, what we have added is one more level of collaboration, in this case, the level of review. So what you see here is an entry in the notebook that was completed by the histology team, but it's important, let's say that it's important for the in vivo team to also review this data. So we have added a sequential option for the review process that can include different teams, different people, and can ensure that that collaboration and around the data is completed. So I know it's a quick overview, but I hope this sparked your interest in learning more about Benchling and the studies in Benchling. And again, please feel free to ask us to give you a full demo with all the details and show you maybe the randomization tool or all the tools in Benchling. Back to you, Shauna.
Sean and Wendy, again, amazing. Thanks so much. And again, if you're interested in taking advantage of these capabilities, you want to reach out to your sales account team to learn more about the new Studies application. Studies product sheet is available to download from the Resources tab. Also, may be helpful to share with others in your org. If you're an existing Benchling customer, your customer success manager can share how to best take advantage of the brand new review process capabilities. Admins and developers can reach out to your customer success manager to see what additional integrations might be helpful. Thank you all for sharing these 2022 perspectives and highlighting some of the great new capabilities for folks to take advantage of. As we look forward to 2023, we are excited to continue innovating on Benchling's R&D cloud. We've got the ability to further connect and automate work across the lab. We're building out more compliance-driven capabilities in biology's modern LIMS, and we continue to lead with even more tools that help scientists model, characterize, design, and analyze complex biomolecules. While we spend some time reflecting on what we're looking forward to in 2023, please check out the poll tab. Get your fingers ready, because we're going to do some cool giveaways here. We would like to share some end-of-year gratitude by sending Benchling theme gifts to the first 100 people who respond with a yes please in the poll. We'll reach out with the best address to send some of our appreciation your way. We hope everyone has a restful holiday and are excited to accomplish more together next year. Here's to ongoing hope and making the magic of science real. Cheers.